So today I'm here to talk to you about an important subject, in my opinion, which is multi-omics and big data in biomedicine and healthcare. So the subject is very important because this is something that is quite recent. We are seeing lots of advances in multi-omics approaches, in multi-omics technologies, along with more efficient use of data and information that we are generating. And specifically, we are generating lots of big data currently. These technologies, these improvements have begun to actually enhance and drastically transform healthcare services in different ways, including the development of individualized, personalized precision medicine. So these technologies, of course, are currently, if you go to any modern clinical setting, you will see these technologies because they are clearly contributing to improvements and to innovations that are employed in many healthcare organizations, in many clinical settings, etc. However, we are still far away from the optimum use of such technologies. This is mainly due because of the complexity of the biological processes in the human organism, and this is making us need lots of tools and lots of solutions to be able to assess and understand and comprehend all of these processes within the human body, within the human organism. So we can actually affirm that no single tool among these can capture a complete and comprehensive understanding of most human diseases and disorders, and hence we need to employ more than just one tool. So again, this leads us to integrate several multi-omics and big data technologies, and this is going to be creating a better, more powerful solution to provide a more complete understanding of human biology and human disease. I think that I am introducing certain terminology here. Most of you, I'm sure that they are already aware of this of these wordings, but I will define all of these in the upcoming slides, of course. So first of all, what are omics and what are multi-omics? Of course, multi means many. So since the process of mapping and sequencing the human genome that began around 20 years ago, so during the early years of the 21st century, we had several new technologies and novel techniques and different processes that actually allowed us to obtain a huge number of potential and possible molecular measurements that we can take within human tissues and even within the human cells. And these technologies, of course, can be applied to different biological systems, and they will enable us to acquire very comprehensive but very easy to grasp snapshot of the underlying biology at a resolution that has never been possible before. So we can go into the smallest of the smallest molecules inside of the human body, inside of the cell. So in general, broadly speaking, we can say that the scientific fields associated with measuring such biological molecules in a high throughput way are going to be called omics. This is the simplest definition that we can give to omics. And as you already know, there are many areas of research that can be classified or categorized as omics, and there are many motivations for conducting omics research and afterwards try to apply what we have found in these research studies, and we try to apply the outcomes of these studies to our clinical settings within our healthcare organizations, or even for research and development departments in other organizations. And one of the most important and most common reasons is actually to obtain a very clear and comprehensive understanding of the studied biological system. So you can have lots of groups of research or researchers working on different types of omics. You can actually find them working on the same process, but they are not working on the same molecular level. So you can, as an example, find researchers that might perform proteomics. We will discuss proteomics and introduce it a little bit later, but here it's just for introducing the overall view. So you can find researchers, for instance, that might perform a proteomics study on normal human kidney tissues to better understand protein activity, to better understand the different functional pathways, and to understand the protein interactions in the kidney. So if we have any sort of deviation from that normal function or from that normal pathway that we label as normal, of course, maybe this is indicating that we have a pathology or a problem with the kidney. So another common goal, in addition to the first one, another common goal of omics studies is to associate the omics-based molecular measurements with the clinical outcome of interest, like trying to understand the survival time for certain diseases. Once someone has certain disease, how long can they live? We can also rely on these studies to assess risks of studied conditions or the response to certain treatments and therapies. And these are going to be helping us to choose better therapies, better treatments in the future. And it's very important to notice here that the motivation and the rationale here are that by taking advantage, the fullest possible advantage of omics-based measurements, there is the potential to develop a more precise, more accurate predictive model or a prognostic model of a particular condition or a particular disease. So we are going to be able to achieve higher levels of precision and accuracy than what we can achieve or obtain relying on standard clinical approaches or conventional traditional clinical approaches. So omics, as I said, are going to be referring to lots of fields of studies, and generally these are going to be ending with the suffix omics. I'm sure that you have heard of genomics, proteomics, but we have also things like metabolomics, transcriptomics, and epigenomics. All of these are the most important ones, but there are actually other omics that are being developed, and we don't have lots of time here to discuss all of these, of course. So, as you know, when we are talking about genomics, we are studying the DNA. When we are talking about transcriptomics, we are studying the RNA. Proteomics, it's the proteins. Metabolomics, biochemical molecules, and these are going to be, of course, leading us to the phenotype, and the phenotype, of course, is going to be the observable, let's say, manifestations of the DNA. So genomics, it's, of course, when you are talking about genomics, we are talking about the genome, and the genome is the complete sequence of DNA in a cell or an organism. And of course, this is going to be generally let's say, something that is stable over time, and this is essentially constant, except when we have certain mutations or certain chromosomal rearrangements, and these are, of course, considered to be as biological accidents. So we are all familiar with the fact that you are going to be digging into the cell to find the chromosomes, and the chromosomes are made up by DNA, of course. So when we say genomics, we are studying the overall genome of someone or of humans in general, and studying genome, including interactions of those genes with each other, and including the interactions of the genes with the person's environment. But actually, when we are talking about environment, there is a sub-specialty that we will introduce later, which is epigenomics. When we consider the transcriptome, transcriptome is studied by transcriptomics. What's the difference? Actually, the transcriptome is the complete set of RNA transcripts that we are obtaining from the DNA. So we have, of course, less genetic information that is translated into RNA from DNA. The transcriptome is going to be including different types of RNA. As you know, we have the ribosomal RNA, messenger RNA, transfer RNA, microRNA, and some non-coding RNA that does not produce any proteins. So as you already know, in humans, it's only around 2% of the genome that is transcripted and represented in the transcriptome as protein coding genes. The rest is not. And transcriptomics is the study of transcriptomes and their functions. So this is why we will have certain differences between studying the genome and studying the transcriptome. So genomics versus transcriptomics, these are a little bit different because genomics will provide an overall overview of the complete set of genetic instructions that are provided by the DNA, whereas transcriptomics studies are going to be focusing on gene expression patterns, and they are going to be actually very useful and very important for a comprehensive understanding of how genes are going to be expressed and interconnected, and how they are going to be influencing certain conditions, certain diseases, etc. So again, we are going to be moving a little bit into more complexity. So we started from the DNA, RNA, and now let's move to the proteins. So the proteins, the overall set or collection of proteins are going to be constituting what we refer to as the proteome. So it's the complete set of proteins that are expressed by a cell, by a tissue, by an organism, and those are going to be different. So if you look into certain organisms or into certain organs or certain cells or tissues, you will not have the same proteome that is formed. And the proteome is actually inherently quite complex because it's not just the result of actually coding the RNA into proteins. You are going to be having some other modifications and some other interactions that happen to proteins. And this is why it's very, very important and drastically transforming our understanding of disease and health by studying proteomics. So proteins can undergo things like post-translational modifications. They can have different spatial configurations, some intercellular localizations. They can interact with other proteins. They can interact with other molecules that exist in the body. So we will have lots of complexity, very high levels of complexity with the proteome. And we are going to be considering proteomics as the large-scale study of proteomes, and we have different sub-areas of proteomics, which are structural, functional, etc. So, of course, you already know that we don't know a lot about the molecular changes that take place in human organs and tissues, especially for certain processes that involve things like aging or things like disease. And we need to have a better understanding actually of all of these. So in order to better understand these changes, we can rely on messenger RNA and protein levels measurements, which can be actually very helpful in this context. So in many studies, you will find in the body of literature that is forming around transcriptomics and proteomics, you will find lots of studies that mainly focus on observing distinctive changes in messenger RNA and protein levels as a function of age or as an outcome of a disease or a health problem. And such changes in both mRNA and protein are going to be associated with different happenings or different occurrences, such as increased immune infiltration. So we have actually dampened our immune response because of these changes, and we have certain decreases in mitochondrial function. So it's very interesting to understand why this is happening. And in this context, proteins actually show a greater extent of change, and they they reveal changes in a wide array of biological processes, including unique organ-specific features. And this is why it's very important to study things and processes and diseases at a proteomic level. So it's very, very enlightening, and it's given us lots of insights about understanding human disease and human and biological processes in general. So this is, of course, especially and even more important as many researchers actually have reported and observed functionally important changes in protein that occur or happen only at the level of proteins. So these are going to be happening at the level of proteins, but they are absent at the level of mRNA. So we see changes that happen in proteins that occur in absence of corresponding changes in messenger RNA, which is actually something that we did not expect. But this shows actually, as I told you, that we have lots of modifications and lots of interactions that can happen for the proteins that are not just the mere result of mRNA. So mRNA profiling, and of course, in general, transcriptomics studies alone are not going to be providing us a complete picture of molecular and biological processes, and therefore examination of changes in proteins is very important. It's crucial. It's essential to understand processes that are not transcriptionally regulated. So as I told you, there are other types of omics studies. We have epigenomics, and quickly it's very similar to genomics, but we are focusing a little bit more on the modifications that can happen in a tissue-specific manner in response to environmental factors, and these are going to be taken into account actually the environment or the context where the person or the population are living, and this is going to be trying to understand the impact of environmental changes or factors in developing certain diseases, and of course whether these changes are going to be persisting across generations or not. We have also the metabolomics, which are actually the study of the biochemical molecules, I'm sorry, and the biochemical molecules can be coming intrinsically, so from inside the human body, or they can be coming from outside. And here we can study so many things like the interactions that can happen between exogenous substances that are coming from outside, and the different metabolites that can be actually produced by these substances. Or we can also look into the interactions that can happen for the different macronutrients, micronutrients. So macro, of course, carbohydrates, lipids, or fats, and proteins. And of course, we can have different things to study here that can be very insightful. In addition, there are many other emerging and new omics technologies that are likely to influence the development of omics-based tests in the future. And as both the types and numbers of molecular measurements continue to increase, we are going to be able to develop better technologies, better techniques, groundbreaking solutions, etc. And advancing bioinformatics and advancing computational biology and computational approaches and relying on some statistical models, we can actually enable improved analysis of omics data. So, and this is going to be helping us have a better understanding of the overall picture. We are going to be having a greater integration of different data types, etc. So to sum it up about omics, as you see, we are getting into more complexity, moving from genome to metabolome. And of course, we are going to be having different things that can lead us to understand the different mechanisms and processes that underlie the production of different molecules within the body. And hopefully these are going to be helping us answer very important questions, like can we improve our understanding of biological and physiological processes of the human organism? What is normal variation within the molecular profile of a normal adult population or within any sort of target population that we want to study? Do lifestyles changes and health behavior interventions and modifications actually impact the data that we are going to be generating? And does this actually influence any sort of the processes that underlie the production of these different molecules within the body, etc. So with the development of omics, we are going to be hopefully able to answer quite clearly all of these questions and even more questions that can be very interesting. So of course, as I told you, it's very important to understand that we need to generate lots of data, and we need to use that data to improve healthcare, to improve our understanding. So the multi-omics are generating data, are giving us information, etc. So we need to use that data. And now we are living in an era where everyone is actually producing some sort of data, and we are living in the era of not just data, but big data. And big data is very important in healthcare. It's a term that we use to describe very big, massive, huge volumes of information that is created by the adoption of digital technologies that can collect patients' records and help in managing hospital performance. These are going to be generally very large and very complex for traditional technologies. So this is why we have developed different technologies to be able to use and to interpret and to analyze big data. You are going to be having databases like Hadoop and other technologies that are very useful in this context. So we used to talk about the 3 V's of big data, but now we talk about the 6 V's. So volume, velocity, variety, veracity, validity, and value. So what are these? So value, this means that the data is clinically relevant. Volume, this means that we have high throughput technologies and we have lots of information and data that are generated, like the measurements of vital signs. Velocity, which is the high speed processing for fast clinical decision support and for actually giving data and information to our clinical decision support systems in order to come up with better medical decisions and in order to reduce the medical errors. Variety, it's about having heterogeneous and different types of data that can be structured or unstructured, which is very challenging from a technological point of view to manage all of these in the same time, but we are able to do it thanks to the technologies that we have developed. Veracity, it means that we have data that is of quality and it is actually going to be able to help us, and it's going to be data that needs to be actually adding something. And variability is going to be mainly about the variations that we can see, like if you are following a certain disease, like currently with the pandemic of COVID-19, you can see variation in the measurements, in the effects, in the number of people who are suffering, etc. So depending on what you are measuring, you will see some level of variation of or variability. So generally, we are going to be generating data and we are going to be using different solutions of hardware to store that data, to save that data. And here, a major concern, of course, is data security and privacy. We don't want people who are not authorized to access the data to do so. So we need to have safeguards for that. It's not actually very interesting now to talk about that. And then we are going to be having the layer that is constituted by software. So we will have here things like data analytics, data visualization, data interpretation, and these are going to be feeding into systems like clinical decision support systems. And these are very helpful for precision and personalized medicine, as we will see. So we are surrounded by an environment that is actually all the time generating and also saving and recording data. We live in a telecommunication environment, and every one of us is actually generating lots of data. And again, here we have, as you see, the different levels of profiles that a person is going to be having. So you can have the DNA gene sequences, so genomics. You can have things like the mRNA. So here we are talking about things like transcriptomics and exomics. We can have things like proteomics that are actually going to be fed by the data that we collect. And according to one estimate that was published back in 2005, mankind created 150 exabytes of data. So it's 1 billion of gigabytes in 2005. You can think this is huge, but in 2018, so merely 13 years later, we generated almost 10 times more of data. So it's not very easy to understand, but you can say that 1 byte is actually 1 grain of rice, and 1 exabyte is going to be actually covering the whole west coast of the United States with rice. So the other consideration here, and I'm going a little bit quicker because we are running out of time, I'm sorry. So health data and health big data. It's a special category of personal data because it's sensitive and it might carry a high risk to rights and freedoms of individuals. So here we have lots of ethical and legal implications. We need to make sure that the data is safe. We need to take care of the privacy and confidentiality concerns. These are things that are very important, and we are going to be using health data and health big data mainly for vital interest of the individual patient, but also for public health interest, like surveillance, etc., population surveillance, etc. And we are going to be having to require explicit informed consent from the patient or anyone we are using their data. And as I told you, we are generating lots and lots of data. We have a huge volume of data in healthcare, and this is similar to other industries. And we have data actually from millions of patients that have been already collected and stored in electronic formats, in the electronic health records, electronic medical records, etc. And we have also the data that is newly generated from self-tracking. So most of you probably have your own smart watches, and you are tracking the number of steps, your heart rate, you are tracking your SpO2, your oxygen saturation in the blood, etc. And these are also data that can be fed into systems and that can help us understand. So we are living in patient-centered healthcare. We want to improve that patient-centered healthcare environment, and we want to make patients more empowered. We want to make health consumers who are not really patients empowered. So people who are using technology for their own health. And this is going to be leading us to generate even more data, and we will have better quality of data again. So let's remember the pyramid. If we have better data, we will have better information, we will have better knowledge. We can apply that knowledge actually to collect even better data, etc. And once we have enough knowledge, enough data, enough understanding and information, we can reach a level of wisdom, so which is very important. So big data can be used as a measure of healthcare also, to understand, are we giving very high quality healthcare? We can use electronic health record data to measure hospital outcomes and patient safety, etc. We can use also big data as a standard, let's say, blanket or standard way to assess the prevalence of patient morbidity by accounting for case mix variation at a hospital by hospital level. So all of these are very important. To conclude, let's quickly talk about the outcome of all of these things: multi-omics and big data and management and use of big data. We are going to be achieving higher levels of precision medicine. So precision medicine is actually referring to the process of tailoring the medical experience to an individual patient. So we are no longer going to be giving the same medication or treatment to everyone who are suffering from the same problem. We are going to be taking into account a combination of their genetics, of their molecular, physiological, and cellular analysis, in addition to their socio-economic or socio-demographic and personal history. So this is not only pertinent to deciding treatment options, but this is also very important in prevention. This is very important in establishing diagnosis and predicting outcomes for the patient and for health consumers in general. So all of these elements actually demonstrate the importance of relying on omics and big data to fathom and to understand and comprehend how to deliver personalized precision medicine. And with the rising costs, of course, of healthcare, we are no longer able to fund mass population screening for diseases. And we know that most patients are not going to be developing these diseases at all. We cannot prescribe costly medication or costly tests that might have no clinical benefits for a given individual patient. This is no longer feasible. We need to tailor and customize the treatment to the patient themselves. So improving precision medicine is going to be offering a solution where we can tailor and customize and personalize a patient's therapy or treatment using their unique data, using their unique genomic and physiological characteristics, which is very important.